want to understand how the brain allows us humans to control the speech and the language that we have that many other species don't have. So we take advantage of the opportunity to study awake human patients undergoing neurosurgery to map different parts of the brain and understand what those parts of the brain do. And we can electrically stimulate part of the brain to temporarily perturb that part of the brain function, and that tells us what that part of the brain does. And then we correlated electrical stimulation findings with temporary brain surface cooling, where cooling will also shut off part of the brain temporarily. And then we measured the speech performance based on perception and different metrics to analyze speech rate and timing. We found differences in speech quality and speech timing based on different brain regions that are both controlling speech motor control. So inferior frontal gyrus, for example, which contains Broca's area, affected speech timing, whereas speech quality was more disrupted when we got more close to motor areas in the brain.
Monday, Tuesday, Wednesday, Thursday, Friday. Monday, Tuesday, Wednesday, Thursday, Friday.
We cool the brain by resting a sterile probe temporarily on the surface of the brain, and we use a Peltier element that electrically gets cold on one side and generates heat on the back side. And we have sterile saline flowing through the back of that to dissipate the heat, and then we measure the temperature beside the probe to know how much we're cooling the surface. Cooling works a little bit differently than electrical stimulation, and that's one thing that we're trying to quantify is how those effects differ. Electrical stimulation probably disrupts a larger patch of brain than it does with the cooling, so that's why we can compare the two techniques head to head. Stimulation has been done for a long time and is known to be a safe and fast and reliable technique to disrupt brain function. Cooling works similarly. Cooling might be safer and less likely to induce a seizure than electrical stimulation, so that would be an advantage. It's not quite as fast or rapid as electrical stimulation, requires some setup, but both can be effective and both can tell us different bits of information like our study is showing. The long-term goal of studies like this and understanding the human brain function is hopefully to restore function in patients that have lost that due to trauma or tumors or strokes. And we need to have first an understanding of how the brain does what it does to develop prostheses and things like that to restore function. So this is one step in that direction.
It's hard to know what exactly the limitations of the procedure are because it's not done on a wide scale at this moment. But some of the things that I could imagine might be the limitation of this is to be able to screen large areas of the brain using this technique. What we have right now with electrical stimulation is we can place a grid over a fairly extended region of the brain and quickly, electrically stimulate these areas and determine which of those regions actually cause speech arrest, for example. With this particular device, since it's limited, as far as I can tell, to one or two devices, you'd have to be able to move these, the device, or there's not, as far as I understand, a mesh of such devices that you can place over the brain and quickly survey the cooling effects. And it also looks like, to me, looking at the graphs, that the effects of cooling appear to take a little bit longer in terms of its effect on speech than electrical stimulation may. That actually I'm not certain about. It's just based on what I looked at and saw in the graphs. Usually stimulation effects are fairly immediate in terms of its effect. The cooling may take time as the tissue cools to see its maximal effect. So I don't know how that will play in practice. Obviously both techniques are invasive, so you would actually have to have this be done during a time of an awake craniotomy, which is when all these patients were tested. I think this is a very interesting technique because it is essentially a novel method by which multiple different hypotheses of brain function can be tested. They mention such as much in their paper. So I think we may see more and more of this technique be studied in a way to look at other types of function, brain function, such as language and memory.